geeks, welcome to another video. So in today's video, we will be discussing those questions that I have posted on my Telegram group as well as it's on my YouTube community page. In case you have not gone and visited those questions, I definitely do recommend you to go and watch those questions, or you can just sail along and look at all the questions that I had posted there and a solution to those questions. I had put a poll as to which questions you were not able to solve, and I've tried to take those questions up and solve them over here. But if you want me to give the solutions to the other questions as well, definitely let me know in the comment section below. And now currently on my group, I am posting questions on immunology. So if you would like to solve those, please rush up to my Telegram group and the link for the same will be in the description down below. So now, without much further ado, let's get right into this video. So the first sum is from biochemistry. I've tried my best to sort it out based on the topic. So the starting few questions you will see are from biochemistry. Then we have a few questions from techniques and finally a few questions on genetics. Okay, I will put up this PDF of the solved questions on my Telegram group as well. Now let us read the first question. It asks you actually what is the fold purification. Now the simplest way to solve this problem is to know the formula. So there is a formula for fold purification. So fold purification is equal to final specific activity upon initial specific activity. It's a very simple formula. Now, as you can tell, you have to have the final and initial specific activity to solve this particular question. So how to calculate the final and initial specific activity? So for the initial specific activity upon the total protein in milligram. Okay. Now from this table that they have given you, you can see that for the first step, when it was the cell-free extract, the activity of the enzyme was 150 and the concentration of protein was 100. So using this, you can say that this is the initial step. So the initial specific activity will be the total activity, that is 150 divided by the total protein, that is 100. So this comes out to be 1.5. Now you can find the final specific activity in a similar way. So final will be 120, which is the total activity after the purification, divided by the total protein, that is 10. So this comes out to be 12. Now all you need to do is simply divide it. So fold purification will be final, that is 12 divided by the initial, and initial is 1.5. So if you solve this, you get answer 8, and that is our option D. Very, very simple. Now for you, I have kept a similar question here. The formula, everything remains the same, just there's a small twist. So you can pause the video and try to solve this, and once you're done, you can replay and check your answer. Okay, so let's dive into this question. So for fold purification, you require the final specific activity and initial specific activity. So the initial specific activity is 1.5 units per ml and 2 mg concentration. So that comes out to be 1.5 units per ml divided by 2 mg per ml. Then for your second, that is the final specific activity, the value is 75 divided by the concentration 100 microgram per ml. Now this is mg per ml and this is microgram per ml. So you need to convert. So let's convert microgram into milligram. So that will be 100. Sorry, we need to convert the mg into microgram. So that will be 1.5 divided by 2 into 10 to the power 3 microgram. Don't solve it, just simply substitute over here. So fold purification will be 75 divided by 100, which is 10 raised to 2 into 1.5, and 2 into 10 to the power 3 will come up, right? Now, if you solve this, it comes out to be 5 2s are 10, 7 2s are 14. So this is 15. This is 2 in, 2 is we've already multiplied, 10 to the power 3 divided by 10 raised to 2 into 1.5. So now obviously if you multiply this by 1.5, you will get 150. So this gets cancelled out. All you have is 10 to the power 3, which is 1000. So the option for this is 1000. Let's move on to the next question now. Again, another question from enzyme kinetics. This is more logical. So it says enzyme X shows an activity on substrate A. So the activity on substrate A is 375. On substrate B is 185. And on substrate C, it is 75. So with respect to substrate A, what is the percentage activities of the enzyme X on B and C? So it's very simple. If you want to find anything with respect to a particular thing, you will write that in the denominator. So now if you want to find percent activity of B with respect to A, you will write B divided by A. So B activity is 185 divided by A, that is 375. And since you want a percentage, you multiply by 100. If you solve this, the answer will come out to be 49%. Similarly, you can calculate a percentage activity of C with respect to A. So that will be C by A into 100. That is 75 divided by 375 into 100, which comes out to be 20%. So this is your option C. Moving on to some of the technique related questions. So this one says a labeled circular single-stranded DNA and a linear short DNA were annealed, okay, to form a product that is shown in one. So this is your the single-stranded circular DNA, and this one is your oligo, that is a short piece of DNA. They were annealed as shown over here, so there must have been some complementarity, and therefore there is hydrogen bonding between these two. So now they are saying a helicase assay was performed using the annealed product, that is 1 and 3 different protein A, B and C. They run a gel and they found out this result. They are asking you that identify a protein which has an helicase activity. So out of these, maybe one protein or maybe a combination of two proteins has an helicase activity. So we have to identify that. Now what an helicase will do is, the helicase will break the hydrogen bonding, correct? So now if it breaks the hydrogen bonding, this structure will get separated. So you will have a separate oligo and the single-stranded DNA. Now, since this oligo is now separated, it is going to run faster, right? Means you will find it at the bottom of the gel. However, if they are remain annealed, they will be near the well. They won't migrate much. So here the bands that you see, these are the bands of the oligo. See, they are traveling very, very fast as compared to the other bands. Now, what would this possibly be? So this will be the linear single-stranded DNA, and this one might be a mixture of the two, which is the annealed product. So let's see what the gel tells you. So in the first well, they have only added B, and you can right away notice that there is no oligo band over here. That means that B is not separating the two. So option B itself we can neglect. Then option C, again you cannot find any kind of band. So option C will also be remain cut out. But D we can't right away cut out because we have not seen in combination. Now in the third scenario, they are adding proteins B as well as C. And again, when B and C are present, there is no separation, no oligo band. Now, they have added A and B together, okay, and here you get your oligo band. So what does this mean? This means that A and B may say one of it has the helicase activity, but in the first well itself we said that B doesn't have the activity. So definitely who will have the activity? It is protein A. Now again, in the next case, since protein A is there, you're getting oligo band. Again, here protein A is there, therefore you're getting the oligo band. Wherever you have A, C, you will have a oligo band. That means protein A has the helicase activity. Moving on to the next one. So this one says protein X exists in two forms, the GTP bound form and the GDP bound form. When this protein was purified, it was always a GDP bound. Okay, so they have a protein X which can remain as protein attached to GTP and a protein attached to GDP. But whenever they purified it, they found that it was always in the GDP bound state only. So as you can see, everywhere you have one thick band. Now what they did was they added three different proteins, Y, W and V. So separately to each they added Y, W and V. Along with that, they added GTP. So your GTP, GTP, GTP. Okay, and here they have added might be only GTP and nothing else, or maybe not even GTP. It's just like a control. Okay, now the results were shown in this way. They're asking what would be the right conclusion. Okay, now they are saying first option, protein Y is a gap for protein X. Now, what do you mean by gap? And you can see another word, GEF. So, gap stands for GTPase activating protein. So as the name suggests, it says that it's a GTPase activating protein. Okay, that means what? It is going to break down GTP to GDP. Then you have GEF, which is guanine nucleotide exchange factor. This guy exchanges GDP for GTP. This usually happens, is done by the RAN proteins, that is RAN GDP, RAN gap and RAN GEF. These are used in G protein coupled signaling pathways. Okay, so now protein A is gap, they are saying. But if it was gap, then you would not be getting a band of GTP, because this so breaks down GTP. So then protein A is not your answer. It says protein Y is a GEF, which is absolutely correct, because it is an exchange factor. In the first place, you have the protein X, which is bound to the GDP. You added GTP to that, and to catalyze this reaction, you have added the guanine exchange factor. So it will exchange this GDP for GTP, and therefore you get a band. You shouldn't look at protein W and B at all, because you are not getting any bands over here. So why waste time? So your correct option is option B. Now, this is a question related to the post-translational modifications. Our proteins are modified after they are made. What kind of modifications are done? The type of modifications done are phosphorylation. Phosphorylation is commonly done at serine, threonine, and tyrosine. Then you have another modification, that is methylation. Methylation is usually done at the lysine and arginine. Then we have acetylation, which is done at GASMB. Okay, glutamic acid, methionine. Then you have your alanine, glycine, etc. Okay, and finally we have glycosylation. Glycosylation are of two types, as we know: O-linked glycosylation and N-linked glycosylations. So the N-linked glycosylation is obviously done at N, that is the asparagine, and the O-linked glycosylation is done at either the serine or the threonine. So now the question here is, consider the following polypeptides that have been analyzed by mass spec. The peptides are generated by cleavage, okay, of a protein expressed in mammalian cell, and it has undergone, okay, it has undergone glycosylation, means it has been added with sugars at either serine, threonine, or asparagine. Phosphorylation, that is, the phosphate group might have been attached to the serine, threonine, or tyrosine, and acetylation, that is at all of these amino acids. What will be the expected positioning of the spectra corresponding to these peptides, smaller to larger? Now, the simple answer to this is option B. Why option B? Because notice there will be a breakage after wherever a post-translational modification has taken place. So asparagine is where the glycosylation must have taken place. Then you have another asparagine over here, then a threonine where you must have had a phosphorylation, and then again asparagine. Okay, so very simple, not much to think about. Next, we have a question based on FRET. To understand anything about FRET, you need to know the principle on which FRET works. So the principle is that the emitted wavelength of a protein is close to the excitation wavelength of the nearby protein. Now we do this in order to determine if two proteins are close to each other in a particular 3D space. Okay, so if a protein is close to each other, then what will happen is, if you have protein 1 and protein 2, which are tagged with two different two different fluorophores. So you have protein 1 attached to fluorophore, protein 2 attached to another fluorophore. Okay, now these fluorophores will have an excitation wavelength and an emission wavelength. Excitation is when it will get excited and transfer its electron, and emission is when it will emit the light, at what wavelength light it will emit. So whenever the two proteins, okay, are close to each other, they interact with each other, the fluorophore is chosen such that the emitted wavelength of 1, that is, if we have the protein 1 ka fluorophore, it will emit a wavelength, okay, a wavelength which will basically go and excite the fluorophore that is present at the second one. Okay, so if only there is an interaction, the second one will get excited and you will get a color. Okay, so now here what they are saying is, in a fluorescent resonance energy transfer FRET experiment, the donor is a cyan fluorescent protein which has an excitation and emission spectra of this and this. So we have a donor which is CFP, cyan fluorescence protein. Its excitation maxima is 435 and its emission maxima is 485. The acceptor in this experiment is YFP, that is the yellow fluorescence protein. The excitation maxima of the yellow fluorescence protein is given as 530 and the emission is given as 545. So if you notice, the excitation wavelength of the donor is 435. So at 435 it will get excited and it will emit at 485. Now look, the excitation spectra of the acceptor YFP is very, very close to the excitation spectra of the acceptor. So if they are in close proximity, then it will definitely, the CFP will, activate the YFP. Now under these conditions, there is a significant FRET observed. What will happen to the FRET signal if the emission maxima of the donor, that is CFP, is made to shift to 475? So instead of 485, they are going to make it emit at 475. Okay, so they are shifting the emission to 475. Now 475 is lower than 485. Now since it will be emitting at a lower wavelength and the excitation spectra of YFP is comparatively very higher, this donor might not be able to efficiently produce a FRET signal when brought in close proximity with YFP. Okay, so the answer is that the signal will decrease. In the other case, if they would have said that 485, they are shifting now to 500, then you would say that the signal is increasing because it is going to emit at a higher wavelength and which is very closer to the excitation wavelength of the other one. Okay, so emitted protein, the emitted wavelength of the CFP is actually shifted to a lower value and this might not be able to produce a FRET, an emission spectra for the YFP. Okay, so this is the answer to this question. Another very simple question, a linear DNA fragment has 3 restriction sites for BAM each one, a restriction endonuclease, and it is labeled at only the 5' end. So at the 5' end of this DNA, you have a label and it has 3 restriction sites. So let's just mark 1, 2, and 3. It is partially digested in such a way that all kinds of fragments are generated. How many fragments you'll finally have which are labeled and how many you will find which are unlabeled? Okay, so let's try and answer this for labeled and unlabeled. So if you do a partial digestion at position 1, okay, at position 1, if you cut over here, see, you will get one of this fragment which will be labeled. So you'll get one labeled and you will get 1, 2. So sorry, you are only having at position 1. So you're only cutting at position 1. So one will be labeled, one will be unlabeled. Now imagine we are only cutting at the second fragment. So this entire one will be the labeled one. So you will have one labeled and then you will have the other one unlabeled, which is this fragment. Now if you do it at 3, this entire thing is a labeled fragment, the large one, and this small one is the unlabeled. So you have one unlabeled. This is at position 3. Now imagine you have cut at position 1, position 2, and position 3 all together. So when cut at position 1, you will get one labeled fragment. Then here will be your second fragment, this will be a third fragment, and this will be a fourth fragment. Now 1, 2, 3, all the second, third, fourth fragments will be unlabeled. So count the total, you will have 4 labeled and you will have 6 unlabeled fractions. Sorry, option D, 4 labeled and 6 unlabeled. Hope this was clear. Now let's move to another very, very interesting problem. Looks very difficult. However, it's the most easiest thing to do. So this is a gel picture given which shows the PCR banding of 2 markers, marker M1 and marker M2. Okay. Now you have 25 different band markers that they have selected. So from this, what you can make out is the total number of DNA samples that are actually loaded in this PCR banding pattern is how much? 25 for M1 and 25 for M2, right? So the total amount of DNA loaded is 50 molecules. So 25 of M1, 25 of M2, right? Now, the next thing that we have to see is where these patterns are differing, because they are asking us to find a linkage distance, and linkage distance ka formula is number of recombinants upon the total DNA loaded into 100. So we calculated the total DNA loaded, it was 25 plus 25, 50. Now we have to see the recombinants. So if you see the binding pattern for P1, it's the same in both, P2, it's the same in both, F1, same in both. This is the parent one, it's not the binding pattern, it's a parent one, parent two, parent, the progeny, F1. Now for the first marker, it's the same, second marker, same, third marker is also the same. It is all the same up till you come to the 11th one. See the 11th one in the M1, okay, for the marker 1, the 11th position is showing a difference as compared to the M2. So here is the first difference, and successively you will look at the 12th one, there also you have a difference. The rest of all the binding patterns remain the same. So if you go to C, you have two markers at which you have recombinants. So now if I ask you the recombinant total, so the total number of recombinants will be how much? So here you have two positions where there is recombinant, and here also you have two positions which there is recombinant. So that will be 2 plus 2, that is 4. Now just for your linkage distance, put in the formula number of recombinants 4 upon the DNA loaded 25 into 100. And this comes out to be how much? Yes, it comes out to be 8 centimorgan. So the answer to this question is 8 centimorgan. Now, next question. So the next question is, a pilot plant treating organic waste in aerobic and continuous mode was running with a constant organic load. It was observed that after 3 days, the dissolved oxygen level has increased from 3 to 4.5 under same operating conditions. Suggest what has been happening to the plant, whether it's running efficiently or not. Now see, one very simple thing that is occurring over here is you have a treatment plant, okay, and this treatment plant is continuously given with some kind of organic matter, so some kind of organic waste that has to be treated. Now see, organic waste to be decomposed needs bacteria, right? So bacteria will utilize the organic waste and convert it and decompose it. Now for the bacteria to carry out any of these functions, it needs to metabolize and it needs oxygen. It needs O2 to basically carry out all of these reactions. So now what ideally should happen is, if initially you have 100 of O2, after a point, the bacteria will start utilizing the oxygen and breaking down this organic waste. So after a point, this value should decrease. A decreased oxygen level will show that the organic waste is being reduced. Okay. Now, in this case, vice versa is happening. Your oxygen level has increased from 4 to 5. This definitely is telling us that the bacteria are not degrading the organic matter because no oxygen has been utilized, right? So this suggests that the treatment plant is not being running efficiently. This is the concept of BOD and COD. Okay. Now here, if you want me to explain a little more about this concept, about the permissible limits, etc., let me know in the comment section. I'll make a small video on this topic. So that's all from me for all of these questions. If you want me to solve the rest of the questions, do let me in the comments below. And if you like this video and if it was helpful, don't forget to like and subscribe. That's it from me for today. I'll see you in my next video. Bye.